So how can vesicles compete? Like they are not live, so.
Yeah, so essentially, you don't have to be live to compete. So competing meaning that, essentially what happens, let's imagine that you have two smaller vesicles, okay? And what happens is, as I was telling you earlier, that the only reason that small molecules like nucleic acids and all, they can go inside, the reason is that these vesicles are sort of leaky, and the leakiness comes from the loose packing. Okay,
Yes.
and loose packing meaning that there is a lot of fluctuations and disturbances in these vesicles. Okay, so it's quite possible that as for example, no, you have to imagine that, so if the size becomes bigger and bigger, and for some reason the packing is loosened, okay, and what will happen is it could be due to the differences of osmotic pressure inside the vesicle and outside the vesicle. Okay, so some of these lipid components can be, sort of in the interface, meaning that, that they might want to come out into the solution. They can want to stay. It's just dynamics. Okay, but
Yes.
if you have some other vesicles which are in close proximity, which are probably more tighter packing, it's very likely that, the from the loose vesicle, the lipid component which is actually coming out, okay, the fatty acid which is coming out, that's sort of incorporated into another vesicle. That can be
Oh,
seen
okay.
as a competition, not a real competition
Mhm.
what you would consider from evolutionary perspective.
Mhm.
Okay, yeah,
And
so, yes.
and sir, one more question that if
Oh,
in the
hold on a second.
okay.
Smriti or Sara, did you start the recording?
Yes, sir. I already
Great,
started the recording.
great, great, great. Yeah, please continue.
Sir, in the Sutherland's experiment, you had mentioned that on shining UV light, only the more stable nitrogen bases remained. But even now, we have mutations in the DNA when UV light is shown for a lot of time. So,
Mhm.
aren't
Yeah.
the
See, so those things are different, right? So if you are talking about a mutation in a DNA, so that mutation essentially leads to I am not sure whether it leads to a change in the nitrogenous base composition, but those mutations essentially mean that an adenine is substituted with a guanine. Okay, or well, it's likely that there are other substitutions, but as we go on to the nucleic acids, you will see that all those mutations have been characterized. There are, it's not that you have A, T, G, C, and U for RNA. You also have certain other chemically modified nucleobases that are extremely well characterized. So I believe that those mutations which happen when you shine UV light, it hovers around those modifications. Okay.
okay?
But in Sutherland's experiment, what they meant is not just the nucleobase, the entire monomer decomposed.
Oh, okay.
Okay.
Okay, sir.
Yeah.
Thank you.
Yeah, no problem. Anything else? Okay, then let's. Am I still audible? Because it's complete silence.
Yes, sir, you're
Yes,
audible.
sir.
Yes, sir.
Yeah. Okay. So this class, we will. It's a pity that I don't have a board. It would have been better with a board. Anyway, so in this class, what we are talking about is the forces that are present in biomolecules. Okay. Now, in this slide, what you see is a structure of DNA. Seen, but some of you may not have seen. But just this is exactly how the. Hold on. Let me put a laser pointer. Yeah. Yeah. So this is the backbone, okay, and this large spheres which you see, these are essentially the phosphorus groups, okay. And so these phosphates and these 5-membered pentagons that you see, these pentagons are essentially the ribose sugar. And you can see that one chain goes in this direction, the other chain will actually essentially goes in opposite direction. Don't worry about the details. When we go into nucleic acids, we will look into that further molecular details of these structures. Okay. So this is how a DNA looks like. There are many other things like a major group, minor group, blah blah blah. And RNA molecule, looks pretty similar, but there are large differences, mainly because you have the additional hydroxy group present at the second position. Okay. But nevertheless, the structure of this DNA and RNA, looks like more simpler than of a protein. I mean, in this protein, here you can probably trace the backbone sort of, okay? By like here in this way and this way. Here, this is very, very nice ordered repeat. But in protein, I mean, it's near impossible to trace the backbone. It's so complex, okay? Now, so now let's try to put it from the perspective of evolution. So if you are to so we were talking about one of the fundamental aspect of life is copying itself. From very simpler terms, you can imagine that it will be much easier to copy a DNA which has a very ordered repetitive structure and composed of just 4 nucleobases as opposed to a protein, because I don't see any order. So and the reason that it's much more energetically demanding, because let's, for the sake of simplicity, consider there are only 20 amino acids. Well, with 20 amino acids, it's very difficult to get an ordered repeat. The chances of mutation, it won't be actual mutation. I would rather call it a randomization. The randomization possibility is much higher in a protein than for a nucleic acid. But at the same time, there is a huge advantage, right? So DNA, if you just consider 4, right? You can do your permutation combination and find out, if you consider a stretch of 20 nucleotides and you have only 4 possibilities. And for a protein, if you consider a stretch of 20 amino acids, which is called as a polypeptide, and you have 20 possibilities, now you can imagine how many diverse sequence of polypeptides you can have as opposed to the DNA or RNA, right? So that's why proteins have diverse functions as opposed to the nucleic acids, right? So it's easy to randomize proteins, and but from an evolutionary perspective, it will be very difficult to have the same protein in a repetitive manner, because there's a lot of energetic cost. If you are to form the polypeptides de novo, meaning that without the assistance of any template, right? And what do I mean by template? You will see in the later classes. All right. So now, why do we have a nice structure of DNA, RNA, and protein? And the reason for these structures and why are they held so tightly, right? The reason are the basic fundamental forces. Now, I am sure that all of you are aware of covalent bonds, and all of you are probably aware of non-covalent bonds, right? So in DNA, you will have the covalent bonds. In all the molecules, essentially, you have the covalent bonds. But as we progress in the next slides and in the next classes, you will see that the non-covalent forces, which are typically very, very weak in nature, those guys play a very, very important role in dictating the structure of these biomolecules. Okay. Now, okay, let's go to the next slide. Is my next slide visible?
Yes, sir.
Yes, sir, it is.
You would have seen this, energy profile, which is a function of distance. Two neutral atoms sort of approach each other from far off distance, right? How do the energy between these two, the potential energy between these two vary. So, now this is essentially shown in terms of two biomolecules, right? Now, one thing that you should always remember presently, but biology is all about molecular recognition. Okay, where if you talk about enzyme catalysis, so enzyme catalysis is fundamentally nothing but you have an enzyme and you have a substrate, but the catalysis will fundamentally not take place if the substrate does not come in proximity to the enzyme, right? It's the same for many other phenomena. For example, transport of whatever molecules across plasma membrane, right?
Hello, sir. Actually, your voice is not coming.
Yes, your voice is breaking.
Hello, sir.
Sir, you are not audible.
Hello.
Yes, sir. Now
Yes,
you
sir,
are
you
Yes,
are
audible.
sir.
audible.
Yes, sir.
Is it able now?
Yeah,
Yes, sir.
you are audible.
Okay, I'm sorry for this. uh, the internet back at my home is a lot more stable than the internet at the institute, which is irony. Can you see the slides, or has it been stopped sharing?
It's
No.
visible.
It's visible.
Okay, great. Okay, you can still see the slide, right?
Yes, sir.
Okay,
Yes, sir.
thank you. Thanks. Yeah. So what I was talking about, that the life is essentially all about molecular recognition. I mean, yeah. So I was at that stage, where we were discussing about active transports and all. For example, I guess I mean, coronavirus is relevant to all of us. And the way this coronavirus guy essentially goes inside our body and causes havoc and perturbs our complete lifestyle, and for some people it's fatal. The reason is the spike protein of the coronavirus. There is something called as a receptor binding domain. Now this receptor binding domain binds to a protein that is present on cell surfaces, which is called as angiotensin converting enzyme 2, ACE2. And it's all about molecular recognition, right? And that's exactly where the process starts. Now, if the ACE2 does not bind to the coronavirus receptor binding domain, which both of which are proteins, this thing, this guy will not come inside our body, will not perturb our lifestyle. So this is the same thing that we have here, right? And it's all about so laser pointer. So it's all about molecular recognition, right? So imagine that this green one is our cell, which has this, not our cell, but this is the ACE2 enzyme that is enzyme, or let's say protein, which is present on the surface of our cell. Okay, and this blue guy is the coronavirus spike protein. Now at a very far off distance, there is no interaction, no attraction amongst them. Okay, but just purely because of random collision, right? Because if these guys are present in solution, which is pretty much the same as in our body, right? You inhale the virus, it goes to your lungs, and on the surface of the lungs, yeah, this virus comes in contact with the cells. On those cells you have this green protein, right? And the green protein comes in contact with the blue protein of the virus, and then what happens? The energy goes down, and the energy goes down because of series of non-covalent interactions that essentially take place between the blue and the green one, right? And it gets further tightened and tightened. Okay, and this is exactly where we come at a specific point. And what is this specific point? You will see at a later slide. And then but the point is, now so this is the energy minimum, meaning that this is the point, or you can say that this is the distance. This is the distance which is the minimal distance of contact between the green guy and the blue guy. If you try to push them further, as you try to push them further, what happens is the energy sharply increases, right? So meaning that it becomes more of a repulsion than an attraction. Okay, and then what happens? Eventually it breaks apart or falls apart. Okay, so it's all about this non-covalent interaction. So essentially, biology, it's all about non-covalent interaction. But covalent interactions are also very, very important, because if you do not have the covalent interaction, you will not have this bean-shaped, this kidney bean-shaped structure. Okay, am I still audible?
Yes, sir.
Yes, sir.
Okay.
Yes, sir. Yes, sir. You are.
Okay. Great. Okay. So now let's. So what happens is, if you are very quantitative, well, so essentially you will see that since this is the introductory course, we will not try to be extremely quantitative, but we will try to be qualitative. For example, some of you might be very interested in how is this interaction energy, right, between this red and the blue. Sorry, the green and the blue. Well, in this case, they are showing between red and the blue. Doesn't matter. So there is a molecule A and there is a molecule B, right? And you can also try to find out computationally how much is the energy between these two interaction partners. There are many biophysical techniques by which you can deduce the energy, how efficiently, how tight is the binding between this molecule A and molecule B. And you can also deduce it from purely pure understanding of physical principles. For example, this molecule 1 can interact with molecule 5, interact with molecule 6, and interact with molecule 7, right? Now, depending upon the chemical environment of the chemical structure of this molecule 1, not molecule 1, but the atom 1 and the atom 5, you'll have some sort of interactive energy between 5, 6, and 7. Okay, same for number 2, number 3, number 4, etc. Now, if you clearly understand the interactions between, or if you have a very good understanding of the interaction between atom 1 and 5, 6, 7, and 2 and 5, 6, 7, so on and so forth, you can actually deduce how strong will be the binding of molecule A with molecule B. It of course requires detailed computational algorithms, blah blah blah, but one can definitely do it. Now, why is it necessary, right? Why are we interested in finding the interaction energy between molecule A and molecule B? Now let's again take the case of coronaviruses, right? Now, one way of preventing the inhibition or preventing the infection of coronaviruses is by making molecules which can prevent the association. So there are different strategies, so it does not necessarily have to be virus, it can be anything, right? Now, so in case of viruses, there are various strategies, but one strategy could be that you prevent the virus to go inside the cell, okay? Because the virus, only if the virus goes inside the cells, it hijacks your machinery and then replicates itself, right? Now, if you prevent the binding of this virus, let's say this blue guy is the virus and the red guy is our cell. If you prevent the association, right, you can prevent the virus to go inside and copying itself. And the way you can do it by understanding how strong is the binding, right? If you can understand how strong is the binding, then only you can design something which is like 5 prime, 6 prime, 7 prime, okay, which can compete. Now we were talking about competition, a while ago, right? Same way, if you add that molecule which is 5 prime, 6 prime, 7 prime, has a similar architecture as this virus, this blue guy, this guy can essentially compete with binding to the virus. But again, you have to design in such a way that you don't take a virus and put on this 5 prime, 6 prime, 7 prime, because then you don't have coronavirus. God knows, you have some other viral infection. But you are to make some sort of a neutral molecule which will probably compete with this, but not go inside your body and replicate itself, right? So that is why understanding these forces is very, very fundamental to life processes, right? You will see later that if we understand these processes, we can understand the architecture or the anatomy of the proteins, nucleic acids, carbohydrates, lipids extremely well. All right. Okay. Now, so far clear? Am I
Yeah.
Yes, sir.
okay?
Yeah.
Okay.
Yes.
Okay. Yeah. I mean, we have not talked, so this is sort of a prelude to what we are going to show in the next slide, but so that you probably appreciate forces. The forces are very, very fundamental in biology. If you do not understand, if you understand biology, or if you understand the forces, you can appreciate really biology, the molecular recognition. Okay. Now, covalent interaction. I'm sure that all of you are aware of covalent interaction. Any idea what is the strength of a carbon-carbon single bond energetically? How many kilocalories per mole? Yes, no?
So maybe 600.
600 what? Kilojoules per mole?
No, 2,400 approximately, I guess, like 2,000, 3,000. Like I meant to say 600, 500, 600 kilocalories per mole.
Correct. Approximately, it's of that order. Yes. Now, it varies depending upon the electronegativity and blah, blah, blah. Okay. And any idea how much is a non-covalent interaction? Okay. Now, okay. Okay, I will go to that in a minute. So, in the next few slides, I'm going to show you some non-covalent interactions. Okay. Now, yeah. So, why are these non-covalent interactions very important? Now, non-covalent interactions are the ones where between two atoms you don't have sharing of electrons. Okay, they don't share electrons. In covalent bonds there is a sharing of the electron. Okay, and in non-covalent interactions you don't really have any sharing of electrons, but you might have certain overlap of the orbitals. Okay, when they are a little attractive. Okay. Now, non-covalent interactions are very, very important. Now, for example, you can imagine. So, so imagine that this green blob is a protein, okay, and there is an amino acid which is sticking out on the surface, and in this case, this amino acid is phenylalanine. Don't worry about the name. When we will go into the proteins and nucleic acids, proteins, we will look into the amino acid architecture and yeah. So what sort of ring functional groups all are present. Now, what happens is, imagine that this phenylalanine is protruded out of the let's say the core of the protein, and it has this sort of a conformation, right? It is it's like a hand which is in a or palm which is right tilted. Now what can happen is there can be a water molecule, okay, and it can come in proximity to this phenylalanine, and it can completely change the orientation of this phenylalanine ring. Now, this change of orientation is very simple. You know why? Because you will see later that, so in this yellow one, can you see my laser pointer?
Yes, sir.
Okay,
Yes, sir.
so this laser pointer is at a carbon atom which is called as a C beta carbon atom. Okay, now this C beta is connected to C alpha, and this C alpha and C beta, this is a sp3 hybridized, so there is free rotation. Okay, now this bond is a covalent bond, but what happens is, because of this collision, you know this is a water molecule which is very polar, and my phenylalanine ring is all composed of carbon atoms and hydrogens, and this is aromatic. It probably doesn't like to interact, okay? And what it tries to do is, because of this collision, collision is more of a qualitative term. It can be some sort of a van der Waals interaction between this water molecule and the phenyl ring, and that is why the orientation changes. And the change in orientation is nothing but just a CC bond rotation. You don't have to make or break a bond here, right? But just simply because of the rotation of the C alpha and the C beta, the orientation of the phenyl ring completely alters. Now, this alteration in the orientation of the phenyl ring can have large consequences to the architecture of this green thing. Here it is not shown, because as we go into the details, you will see that what sort of alterations can this guy have. But I can just tell you briefly that, imagine that this phenyl ring is engaged in some sort of a stacking interaction with another phenyl ring here. Okay. Now, are you familiar with a stacking interaction, pi-pi stacking interaction? If
No,
you have
sir.
heard, say yes.
No, sir.
If you have
No,
not, no, no, don't worry.
No,
Don't
sir.
worry.
Yeah.
Don't worry. Don't worry. It's okay.
No, sir.
Don't worry. So this is maybe I will stop sharing and at least turn on my video. Can you see me?
Yes, sir.
Okay. So can you see my hands? Can you see my hands?
Yes.
Okay.
Yes, sir. Yes.
So imagine this is a phenyl ring and this is another phenyl ring. Okay. All right. So what happens is, one phenyl ring can actually come in very close proximity to another phenyl ring. So they are sort of parallel. Okay. But because these phenyl rings, you have an electron cloud which is revolving around, right? Because this is aromatic, right? And that is why when you draw a phenyl ring, for example, you can draw a delta negative, which is essentially going around all the 6 carbon atoms. Okay, so they don't, two phenyl rings, they cannot approach each other from face to face. But what happens is, there is a little offset. Okay, meaning that there is one phenyl ring here, and the other phenyl ring can come, in a parallel orientation. Okay, but there is a little offset. Why is that offset? The offset is fundamentally because the phenyl rings are connected with the CHs, right? And the hydrogen, it is electropositive. So what happens is, there is some sort of attractive interaction between the negative charge, which is sort of delocalizing on the phenyl ring, and the hydrogens, which are essentially sticking out from the phenyl ring, another phenyl ring, and they sort of attract each other. Okay, and since they attract each other, two phenyl rings can be in this kind of orientation. Okay, this is called as a face-to-face interaction. It's also possible that one phenyl ring can also interact with the other phenyl ring in a T-shaped manner. Okay, So there is the hydrogen atom, which is not the hydrogen atom, but the CH, because you have, it's a hexagon, right? On the tip of this, you have the CH, and in this ring you have the delocalized electron cloud. Okay, the CH can come in very close proximity to other one. Okay, so these interactions are called as stacking interactions. Now you probably would appreciate these are not full-blown negative or positive charges. These are partial positive charges. These are sort of dipoles, right? And this is like a dipole kind of interaction. And this is what is your van der Waals interaction, okay? The energy between this kind of interaction, which is offset stack or T-shaped interaction between two phenyl rings, is very, very low. But what can happen is if you have that non-covalent interaction initially, imagine that yeah, so again take the example of a coronavirus. Okay, so imagine the spike protein, which essentially, or the receptor binding domain of the spike protein of the coronavirus, which comes and binds to our cell, has two phenyl rings which is involved in the binding. Recollect that figure where I showed you 1, 2, 3, 4 interacting with 5, 6, 7, right? Imagine 5 and 6 are two phenyl rings which are sort of offset stacked, right? Now, if there is a non-covalent interaction which alters the orientation of this phenyl ring, what will happen is the orientation of this phenyl ring either will stay constant or that will change. But because of this, the change of this orientation of the phenyl ring, it can completely prevent the binding of the receptor binding domain to the ACE2. Okay, and that can completely hamper the binding. So you can imagine how this non-covalent interactions, such very small interaction, can be so important in determining biology. Okay, so in this case, we are talking about the infection, right? It can be signaling processes also. It can be evolution. It can be anything. Well, that's why we say biology. That's why these non-covalent interactions are so very, very important. Okay, could you still hear me?
Yes.
Yes.
Okay.
Excuse me, sir.
Yes, sir.
Okay.
Excuse me, sir.
Yes. Tell me while I share my screen.
Sir, the two phenyl rings had a non-covalent interaction.
Oh, hold on. Hold on. Let me start sharing my screen. Yeah. Yeah, tell me. Yes, I can hear you.
So the two phenyl rings had a non-covalent interaction,
Yes.
and there was a collision, and it
Yes.
altered the orientation.
Yes. This is what we are seeing here.
Sir, but in the second slide, sir, we had different phenomenon or something. In that we had a blue and a green protein. They were interacting by a non-covalent interaction.
Are
Or
you
this.
talking about, can you see my slide?
Yes, sir.
Are you talking about this blue small sphere?
No, sir. The last green and blue.
This one?
Second slide. Second slide.
Yes.
No. So that energy, and yes, this.
I am here. Yes. Maybe there is a slow streaming. Yes. Tell me.
Sir, in the positive energy graph, in the positive energy part,
Yeah.
these two can be considered as the two phenyl rings, or is it the collision?
Correct.
So it is the collision or the two phenyl ring interaction.
No, not necessarily.
Sir, the interaction which was a collision alters
Sir, if I may
the
pitch in,
orientation.
uh, I think there are multiple types of non-covalent interactions, and I think sir is explaining different types of them, if I'm not
You're
wrong.
right. You're right. Yes, I'm trying to explain different types of non-covalent interaction. But anyway, go ahead with your question. Okay, now I understand. What you're saying is collision means it's an energetically unfavorable phenomenon, so which will be typically considered in this domain. Is that what you're trying to say?
No, sir. I'm saying the collision was a non-covalent interaction, but the distance cannot decrease in that. Hello?
Distance cannot be, well, yeah, so hold on for a few minutes, okay, until I explain that part, okay, and then you can either your question will be clarified or you still might have a question, then you can ask me, okay? All right, great.
I think, sir, this one represents enzyme substrate interaction.
Can be
This
anything.
blue and green one.
It doesn't have to be enzyme substrate, it can be anything.
No,
Okay?
it can be anything. I mean, it represents that type of interaction, and the last one represents a different type of interaction, I think.
I mean, this
Yeah,
might as
no.
well
So,
be some.
I mean, if you go to the textbook probably talks to you about the enzyme substrate interaction. But what I'm trying to explain is, well, enzyme, why they explain enzyme substrate, because this is 100 years old lock and key mechanism, okay? And if they typically say enzyme substrate, it hits to everyone that, oh yes, enzyme lock and key. But there are something which is called as protein-protein interaction. This can also be visualized as a protein-protein interaction. All right. It can be anything. It doesn't have to be enzyme substrate. Even if it's an enzyme substrate interaction, if you do not consider the catalytic step, it will be a protein-protein interaction. All right.
Okay, sir.
All right. Anyway. Yeah. So that's what I was saying. So this is so my whole point is to explain that non-covalent interactions are very, very important because they lead to conformational change, which is written here, conformational change. Because it's not important that you always have to break a bond to bring about large changes. Subtle alteration in conformation of biomolecules can lead to very, very large, spectacular changes. Okay. All right. Now, in this case, so here we are talking about some sort of a collision where there is no attractive interaction between a phenyl ring or any other amino acids which are in close proximity. But the same thing holds true if you have a ionic interaction. For example, this residue here is a glutamic acid. Don't worry about the name. We will see what glutamic acid is. And this residue here, at the terminal of which there is a guanidinium group, it's called as an arginine. These two are amino acids. Now, at physiological pH, okay, which is 7.4, this arginine is positively charged. The terminal of this arginine is positively charged, and the terminal of this glutamic acid is negatively charged. Now, if this glutamic acid side chain, this is called the side chain, is in proximity to the side chain of the arginine, meaning that this negative charge and positive charge are in proximity, there is an attractive interaction. Okay. Now, what happens is, now that interaction is very strong. In few minutes, we will look into the slides and again qualitatively compare the energy between these interactions. But it's quite likely that if you have a water molecule which comes in proximity, you can potentially disrupt this interaction. Okay, so this is again another kind of non-covalent interaction, which is an ionic interaction. Okay, all right. Now I'm sure that
ফ্যানটা চালিয়ে দাও ধুলো উড়ুক ধুলো উড়ুক আমি পা
can
টিপে
you
গরমে
please
থাকবো
turn
ধুলো
off
উড়ুক
your
শ্বাস
mic?
নেবো ধুলো তুমি দাও ওরা
Thank you. Yes. So so this kind of interaction, I'm sure that all of you have at least read about the interaction between a positive and a negative charge. What is it called? How can you deduce the force between a positive and a negative charge?
Coulomb's law.
Coulomb's law. Correct. And now if you have a water molecule which is coming in proximity, now can you tell me a phenomenon, or can you use this water molecule, or is the water molecule interaction somehow represented by the Coulomb's law? What do you think?
Yes, the dipole-dipole interactions.
No, no, no. So what? Yes, yes, it's a dipole-dipole interaction. But what I'm asking is,
Dielectric.
so is this, so the positive and a negative charge is an attractive interaction, right?
Yes.
Now what happens is, if you have a water molecule which is coming in proximity, the force between these two, the attractive interaction between these two sort of weakens out. Is this phenomenon captured by the Coulomb's law?
Dielectric effects.
Correct.
It's a
Yes.
dielectric effect.
Correct. Yes. Which is called as the relative permittivity. Okay, the epsilon.
Yes.
Right?
Yeah.
So that epsilon is actually capturing this phenomenon, right? Which is for water, it's like 80.5, 81, whatever. Okay. Now you can imagine if you have a positive and a negative charge. Okay. Maybe I can go to the. Where is that? Yeah. Yeah. I will go to this slide. Can you see my slide?
Yes, sir. We can see a protein and 3 angstrom written.
Correct. Good. Good. Yes. So, so this is exactly what is happening. Now, I have jumped a few slides. Don't worry, I'll go back again. But in vacuum, for example, if you have the same positive and the negative charge, where you don't have the epsilon is close to 1. Let's say it's just 1, okay? Or minuscule in vacuum, okay? The interaction energy between this, you can probably calculate when these two charges are separated by 3 angstrom is 500 kilojoules per mole, or whatever, maybe 125 kcals per mole, okay? Whereas if you have the same charge separated by 3 angstrom in water, of course you have the dielectric constant of water, right? Now the dielectric can essentially shield this interaction energy, and then the interaction between these two drops to 6 kilojoules per mole, or maybe 1.25 or whatever kilocalories per mole. Now this is something that takes place in a protein, right? Now why am I talking about protein? Because as we go later in proteins, we will see that proteins have, well, the interior of the protein, proteins are, you can consider protein as this a sphere, okay? Now, I mean, it's a very simplistic representation of a protein, but you can imagine it's some sort of a hollow sphere. The interior is, and this hollow sphere is submerged in a bucket of water, okay? So imagine, this is in the water. Now what happens is, on the surface of this ball, you can consider a tennis ball, maybe, okay? So on the surface of this tennis ball, you can have this positive and negative charge, okay? Now the interaction between the same positive, the same glutamate and the arginine, okay? It will probably be very, very low, okay? But the same interaction, if you consider the same interaction between the glutamate and the arginine, if they are present within the inner side of the ball where there is no water, there will be a very strong attractive interaction. Okay, and that's how you see that the same coulombs, the same salt bridge or ionic interaction, depending upon the environment where they are present, right? It can be either very, very strong, and such strong interactions can maintain the rigidity of the core of a protein, whereas the same interaction, when it is present on the surface of a protein, because of the water molecules present there, this interaction can be extremely floppy, okay? And that can be broken by some sort of a non-covalent interaction between a water molecule. It doesn't have to be water molecule. It can be other charge interactions also. All right. So yeah. So that's why I just wanted to put things in perspective so that you might appreciate that how important are these non-covalent interactions, and they act differentially. They act differentially in an environment specific manner. Okay. All right. Now we can go back. Yes. Yes.
While considering the dielectric interaction
Yeah.
between the two molecules, but right now we are seeing that the distance between them is 3 Armstrong, which is very small or is like comparable to the size of the water molecule. Does this have a varying effect on the dielectric, like a weakening of the strength of the bond as compared to, when we consider bulk of water? Because in bulk of water, we can easily see the, what do we say, the dielectric effect, but is it the same when we talk of small distances of the order of the size of the molecules?
I didn't quite understand your question. I mean, can you try to rephrase it? I
Yes,
didn't
sir.
understand the bulk water and the distance. I mean, are you talking about that how important is this distance, or how important is the proximity of the water molecules to these interactions?
So generally, when we talk about the dielectric effect of water, we usually have some bulk of water, but in the distance which you said was 3 Armstrong, we
Yeah.
cannot have more than 1 or 2 molecules of water present over there. So does this have a varying effect as compared to what we normally would see in like bulk of water?
Well, I mean, uh, well, I mean, okay. So what I mean to say here is, so let's say the interaction is 3 angstrom, because what happens is, this is again a non-covalent interaction, and you can imagine the same kind of graph between when two hydrogen atoms approach each other, right? At certain distance, you have a very attractive interaction. Okay. Now, what I mean to say here is you would probably see the same interaction in bulk water, or if there are just few water molecules at the surface of the protein and the water molecules, because the water molecules do not necessarily have to go in between these 2 point charges to nullify that interaction, because you have to consider few things. Now, see, what happens is, if you have a positive and a negative charge, if there are water molecules, there is something called as a solvation. Okay. Now, what happens is, I mean, well, it can be very well understood from the slide that I have here, right? So, for example, imagine a water molecule is this yellow sphere. Okay, it's a neutral. Well, here it's not an atom; it's a molecule. Okay. Now, if you have a positive charge, what happens is you have an induced kind of dipole, right? You can imagine, you have the water molecule where there was no positive charge, but when the water molecule comes in proximity to the positive charge, there is an induced dipole, right? And what happens is, because of this induced dipole, even if you have a negative charge around, there is an attractive interaction between the positive charge. So in our case, there is an arginine residue. So this arginine residue will start interacting with the water molecules that are present here. Okay. Same for the negative charge. If we have a negative charge in proximity, the negative charge will induce a dipole into the neutral water molecules which come in proximity. Then the negatively charged glutamate will start interacting with the water molecules which are in proximity to the glutamate. Okay. Now, effectively, what happens is, now the positive charge is interacting with a couple of water molecules in proximity. This negative charge is interacting with a couple of water molecules in proximity. So the net interaction, or the net force with which, or the net attractive force between the positive and the negative charge here is reduced, right? So they will weaken out, as opposed to if you don't have these water molecules, right? If you don't have the water molecules coming in proximity, then you won't have this induced dipole kind of effects, right? And whatever electrostatic interaction you have between the positive and the negative charge, it will be maintained. So now, so you probably understand that even though there is a 3 angstrom distance between the positive and the negative charge, the water molecules don't actually have to come and sit inside, they can very well surround the positive charge, and not very well, they will actually very well surround the positive charge and the negative charge and effectively weaken this interaction. Does it answer your question?
Yes, sir. Yes, sir. Thank you.
Okay, yeah, great. Okay, so yeah, so this is exactly what's I mean, okay, it was great. It was a great question. It actually explained the slide very well. I mean, I wanted to explain from a different perspective, but probably you understand it better now. Okay, now, so what happens is, you have a neutral atom. These two neutral atoms are not really neutral. So what happens is, why okay, where is this? Yeah, why do we see this drop in energy between two neutral molecules? Because no matter how you decompose, a neutral molecule is actually has a fluctuating positive and a negative charge. Now, when two neutral molecules come close to each other, of course, there is a dipole induced dipole kind of effect, and that is why you have a positive charge orienting towards the negative charges, right? And that is why the interaction when at very far distances, they're pretty much neutral. There is no attractive interaction. But as they approach towards each other, there is a dipole-induced dipole kind of effect, and the energy drops. But beyond a certain distance increases. Now, what is this distance, right? So now we come at this slide where you have two neutral atoms, two dipoles, which are at very far off distance. There is no attractive interaction whatsoever. But as they approach each other, right? As they approach each other, because of this mutually induced dipole kind of effect, they attract. Now, they essentially go to some specific distance, because in x-axis we have the distance, and in y-axis we have the energy in kilojoules per mole. They attract each other, but then further, if you try to push it, okay, because, well, this picture holds true if you consider atoms as sort of hard spheres, okay? Because if you try to push two hard spheres, imagine you have two soft balls, right? You bring the two soft balls close to each other, because of the simple electrostatics, they will probably attract each other. But if you try to push them, what happens? It's essentially, at this part, you try to imagine that, you are trying to push two soft balls close to each other. You can probably push them, but there is enormous collision energy because they are actually coming very close. They're trying to overlap with each other, and that's why the energy rises. And the energy, of course, rises, which is much higher than the attractive interaction or the attractive dipole or the electrostatic interaction kind of interaction between them, and it steeply increases. Okay, and this distance, the distance at which two molecules can come close to each other, is called as the van der Waals distance, right? Which is essentially the sum of the van der Waals radii of this atom and this atom. If you sum these two distances from the center, this is the distance at which they can come in proximity. Beyond this distance, beyond the sum of their van der Waals radii, you try to push them, the energy is going to sharply increase. Okay. All right. Now, there is some sort of a approximation here that is not really true all the time. Okay. Because I spoke to you about non-covalent interactions. Okay. Now, there are many non-covalent interactions, and the way people actually identify those non-covalent interactions are by determining whether the sum of their van der Waals radii, or let's say that, so if you are to determine if there is a van der Waals interaction between two atoms, what you do is you calculate the distance between these two atoms. And if the distance between two atoms is less than the sum of their van der Waals radii, then it means that these two atoms are actually attracting each other. Okay, and in this way, people have identified many non-covalent interactions. You will probably read later in your uh, in your chemistry studies. There are some interactions which are called as CH pi interactions. Okay, you will read about something called as N2 pi star interactions. You will read about cation pi interactions. Many such interactions where people have actually identified those by determining how close these two atoms are. Okay. Now, in a classical scenario, as I said, they're considered as hard sphere, but these two hard sphere will come only as much their van der Waals, individual van der Waals radii allow them to, which is this dotted line. But in many cases, it turns out they come in close proximity if their van der Waals radii, and their van der Waals radii is much less than these dotted lines. Okay, and based on this, you can say that, oh, these two atoms are actually attracting each other, and since they are attracting each other, there is overlap. There is this kind of overlap. So it's not necessary this overlap means repulsion. This overlap can also be attraction, meaning that this graph will not necessarily come and dip to their van der Waals radii. It can also go down, and it can go deeper. It can be shorter than their van der Waals radii. The energy will further dip, and beyond that, then the energy will increase. Did you understand what I tried to say?
Yes, sir.
Followed?
So I think
Yes,
there is
sir.
a doubt. Sprithu,
Is there a question? Sorry, I could not
Sir,
hear
is
you.
it possible? Excuse me, sir.
Yeah.
Sir, the van der Waals radius can be greater than the sum of, sorry, the radius which we are talking about
Yes.
can be greater than the sum of the van der Waals radius also. Their individual.
No problem,
Even
greater,
then
of
the
course.
energy would have. Hello?
Yes, greater, no problem. Greater is no problem, but smaller is something that we need to look into carefully. That's what I mean. If it's smaller, then it means that there is some sort of an attractive interaction which cannot be explained by the classical hard sphere model.
If the radius is smaller than their individual, then even then there's a possibility that the energy would decrease.
Yes, correct.
And the distance when they have the minimum energy, will consider that distance as the van der Waals radius then, or any other interaction will consider that as?
No. The van der Waals radii is typically, that is known. So, for example, here. Okay. So, do you see this table?
Yes, sir.
Yeah. So, for an isolated atom or so, or maybe from diatomic molecules, diatomic elements, or whatever, the van der Waals radii of oxygen, chlorine, nitrogen, all the elements are known. Okay. Now, what I am saying is, imagine, so the van der Waals, if you have a carbon and an oxygen, if you consider the hard sphere model, you will see that the closest proximity that a carbon and an oxygen come will be the sum of these two, meaning it's 1.5 plus 1.7, it's 3.2. Okay.
Yes, sir.
But in some cases, it is observed that the distance between carbon and oxygen is shorter. Okay, well, we are not talking about a carbon-oxygen bond. Okay, we are talking about carbon and oxygen which are not bonded, which are not covalently bonded. Okay, in those cases, it is observed that if you can experimentally determine the coordinates of carbon and oxygen, and there are many techniques. One such technique is X-ray crystallography. X-ray crystallography. By doing X-ray crystallography, you can determine the coordinates of carbon and oxygen in proteins, for example, and you can see that In some cases, the distance between a carbon, imagine there is a carbon here and an oxygen here, which are not covalently bonded. And if you just find the distance between the carbon and the oxygen, it will be less than 3.2 angstrom. Okay.
So we'll call that interaction as a van der Waals type only. We'll not consider that as a covalent.
Yes, no, it will not be covalent because there is no bond. If there is a covalent bond between the carbon and oxygen, the distance will be much shorter, right? It will be around 1.4 angstrom or 1.5 angstrom. Carbon oxygen bonds, like for example in CO2, depending upon the hybridization, of course, right?
Yes, sir. Yes, sir.
But what I'm talking about is, one carbon in proximity to another oxygen which are not bonded. Ideally, the distance will be the sum of their van der Waals radii, 3.2. But in many cases, it is observed that the distance is much shorter. So that means, if the distance is shorter, that means that there is an overlap. So this overlap, what you are seeing here, it is not always repulsive. It can be attractive also. Okay?
Clear, sir. Clear, sir.
All
Thank you,
right.
sir.
Thanks.
So, sir, in those cases, the energy graph would dip lower, right?
Yes, absolutely.
Okay.
Absolutely. The energy graph will not be here. The energy graph will be lower and it will be further on the left side. The distance will be shorter, of course.
Yeah,
So in
thank you, sir.
other cases, like non-covalent interactions and non-van der Waals interactions, like say dipole or ion dipole interactions, the energy will dip further low, but the distance will remain the same.
No,
right?
distance will not remain the same. Distance will reduce.
In dipole or ion dipole as well.
Yeah, of course. I will show you in a minute.
Okay.
Distance will definitely reduce. Okay, so
Sir, I had one question.
Yes.
So my question is that here we can see that the thermal energy at room temperature is quite a lot greater than the deficit in energy caused by the van der Waals energy by this. So will the thermal agitation break these van der Waals interactions very frequently then?
Oh, that's a very good question. Yeah. See, thermal energy is essentially the energy between molecules because of random collision. Okay. Now, in this graph, it shows that the thermal energy is around maybe minus 2.5 kilojoules per mole. Okay. All right. Now, this, and whereas if you consider the van der Waals interaction energy here, in this case, it's minus 1 kilojoule per mole. Okay. It's typically in that order. So it means that the random collision, the thermal energy at room temperature, let's say 300 Kelvin, okay, it's far higher, okay, it's far higher to disrupt the van der Waals energy, right, or van der Waals interaction. Then why is that meaningful? Why are we studying about this if it's not really meaningful in biology? Okay, the reason is, in many cases, these, that's what so that's why so let's imagine in the context of this. So those van der Waals energy, now in this case we are talking about a real electrostatic charge, real charge, okay, but you saw that these are this also can be considered in the context of dipoles, because there also you have transient charge, fluctuating charge, but essentially it's charge. Now imagine this is not an electrostatic interaction. These are just dipoles. Now if this dipole, okay, is in water, okay, where the chances of collision is much higher as opposed to in vacuum, right? This 1 kilojoules per mole will not really hold true. Probably on the surface of the protein, maybe it is much lower, but in the interior of a protein, this energy let's consider that this energy is just 1 kilojoules per mole, but then this 1 kilojoule per mole energy between these two atoms stays put because there is no thermal energy in vicinity to break this interaction. It's an let's consider a scenario, okay? And the other thing to consider is many of these van der Waals interactions, they are not just single interactions. there are cascade of such interaction. Okay, when we study about alpha helices, I will give you one example of such interaction. Okay, you will see that those interactions, there are a series of those interactions, and those, if you add them up, they will be much higher than the thermal energy at room temperature. And then you probably can appreciate, oh yes, then that will have some meaningful consequences or repercussions on the anatomy of biomolecules. Does it, I mean, you probably, does it answer your question, or
Yes, sir. Yes, sir. Thank you, sir. Yes. Thank you.
okay. All right. So now, yeah, one such example of this van der Waals interaction is, it's again taken from the textbook, is the way that lizards or geckos move on surfaces. Because apparently it turns out that there were many theories to explain how the geckos actually move up. But if you look into the feet, the feet of these geckos, there are rows of setae, and each setae has, as you can see the scale here, this is 75 micron, and these rows of seta, if you zoom in, now we are in the 1 micron range. There are sort of millions of these small tiny guys called as spatula, right, which are really sub-micron in length. Now, no matter how rough a surface is, right, I doubt that our eyes can see 1 micron length. Our eyes do not have such resolution. But if you take a microscope, you will see that no matter how rough a surface is, we can always find surfaces, or surfaces which are very smooth in the range of 1 micron on the surface of a wall, where there can be very nice van der Waals interaction between this spatula and the wall, and there can be, as I said, that one spatula is not enough. Since there are millions and millions of spatula, this spatula can very tightly grip the wall, purely van der Waals interaction, right? One energy, you can consider that one to be, like 0.25 kcal per mole. If there are thousands of those, you can actually imagine that how much energy would that be. And because of this van der Waals interaction, these guys can actually climb up the wall. Okay? Very simple. But because of cooperative interactions, thousands and millions of those van der Waals interaction, acting in one go, they make real. And this is exactly what happens to protein structures. You will see later that hundreds of those van der Waals interaction act together, and that is why, although they are not ionic, although these individual interactions are not covalent, they don't have such huge energetic component, but minuscule energetic component. But the reason is there are so many of them acting together, they make real sense. They make real phenomenon, real, so it's, you observe a physical consequence of such weak interactions. All right. Now, yeah. So, as I was telling you, that we have this. This table is important. I'm sure that you would have seen this table maybe in your chemistry textbooks, or you will see it in your chemistry textbook. You have the atoms, and you have the van der Waals radius, and at the same time, electronegativity is something we must appreciate, right? And the most evident electronegativity, or the consequence of electronegativity, is the hydrogen bond that we have in biomolecules. Okay, so you will see that the most electronegative atom in Pauling electronegativity scale, well, which typically we find in biomolecules, is oxygen. If you just talk about electronegativity, probably fluorine will be the highest. Okay, so oxygen, you have a van der Waals radius of 1.5 angstrom. You have, in the electronegativity scale, you have 3.4, and yeah, so different atoms which are typically found in biomolecules. You have the oxygen, carbon, hydrogen, nitrogen. You also have phosphorus, like in DNA, you have the phosphorus, and sulfur is also, if not so prevalent, but you do have sulfur. As you will see that there are certain amino acids which contain sulfur. Chlorine. Well, there are not just chlorine, you do find fluorine also. Not so not so prevalent in biomolecules, but they are found actually in natural biomolecules. Probably, fungi and algae produces biomolecules which have several halogen atoms. Okay. Okay. So yeah, so you have the typical van der Waals radii and the electronegativity. Now, so this is again, pictorially describing that arginine and a glutamate, and within a protein, and how arginine and glutamate can make real sense when they are present, on the attraction between these two are very, very important in maintaining the structure of the proteins, right? Why am I saying proteins? Because arginine and glutamate are amino acids. Okay. Now here we have so just like the van der Waals interaction. So this is the van der Waals between two neutral atoms. This is the energy diagram, and if we have an ion pair, you see, as one of your colleagues asked, so you can see if you have an ion pair, of course the energy is much higher between them. Okay, and of course the distance is also shorter. Okay, so they go beyond the van der Waals radii. Okay, and then, but again, obviously there is a limit how much you can push, right? Beyond a certain limit the energy steeply increases. Now, yeah, so this is what I was explaining, in terms of the electrostatic interactions, and how water can actually shield electrostatic interactions. And shielding of such electrostatic interactions is very important. You can imagine that, for example, if you do not have water molecules, and if you have a protein which is rich in negative charges, if you have a protein which is rich in positive charges just on the surface, then this just purely because of electrostatic interaction, this negatively charged protein will attract the positively charged protein, and it will lead to attraction between two proteins, and eventually aggregation and all kinds of crazy things, right? They will all attract each other, and will form a blob. But if you have water molecules, the water molecules nicely shield the charges, right? They nicely solvate the charges because of the solvation, right, of the positive charge and the negative charges, because of the formation of hydrogen bonds. you know a protein which is rich in positive charge and a protein which is negative charge. In this case, you have a protein which has both positive and negative. But imagine a protein which is rich in positive and a protein which is rich in negative. They will not come and attract each other and just form a blob and neutralizing the activity of both the proteins, right? That's why water is so fundamental to life, because of its dielectric, the shielding property, okay, the hydrogen bonding property. All right, and that is why so the hydrogen bonds are very important. Now, I'm sure that all of you know about the hydrogen bonds. Yes or no?
Yes, sir.
Okay. So yeah. So for example, here and of course, you can probably appreciate that hydrogen bond is important, or you have a hydrogen bond if the atoms between a bond is polarized, meaning that there is separation of charge, right? For example, this is a peptide backbone. Don't worry what a peptide backbone is. This is oxygen and this is a carbon, right? This is a C double bond O. Now, since you have a large difference in the electronegativity between the oxygen and the carbon, right, there is some sort of a dipole. It forms a dipole, right? The same from this is a nitrogen and a hydrogen, okay? And the same thing holds true since you have a nitrogen and a hydrogen, which are there's a large difference in electronegativity, right? It's shown here. For example, hydrogen is 2.1 here, and the nitrogen is 3, right? There is a large difference. Same thing for the oxygen and the carbon. There is a large difference. But if you just consider, for example, a carbon and a nitrogen, right? So this is 2.6 and 3.0. You don't have a large difference between the electronegativity, although you do have a difference, right? And because of this large difference in electronegativity, you have creation of this partial dipoles. And since you have the creation of partial dipoles, you have the formation of hydrogen bonds. Now, one of the fundamental character of hydrogen bond is the presence of a hydrogen in a hydrogen bond, right? Because hydrogen is the most electropositive element that we can imagine, right? So it's really electron deficient. Since you have electron deficient center here, that is why there is the positive, or there is an attractive interaction between a negative charge and a negative charge through the hydrogen, right? Now, but if there was no hydrogen here, you would not have, if it was just a nitrogen and the oxygen. Of course, although there is a difference in electronegativity, right? So, this will be 0.4 or so, but there won't be any hydrogen bond. Okay? So, this hydrogen bond is very, very important. And it turns out that, well, the consideration of a dipole is too simplistic for a hydrogen bond. Okay? You will probably study later that hydrogen bond is really an approximation if you just consider hydrogen bond as two dipoles. Here you have a dipole and here you have a dipole. They come in proximity. It's not really dipolar interaction. In true sense, there is actually an orbital overlap, and since there is an orbital overlap, I mean. So there is not say no orbital overlap. Let's say there is a quantum mechanical nature of this interaction. Some people also believe it's an electrostatic kind of interaction, and since there is some sort of an overlap, that is why the distance, or not the distance, but the orientation of this NH and the CO is also very important. Okay, so.
Sir, I apologize to interrupt, but I think we are over our class time.
Oh, thank
So,
you. Thank you for telling me. No need to apologize, please. It's very
alright, sir.
important you tell me. Okay. So
Yes.
I guess the other classes began. If someone has a burning question, please. What is the time? Oh my God! It's 12:24. Oh, I'm sorry. I'm really sorry. Okay, you guys carry on. Okay, we will describe in the next class. If you
Thank
have,
you, sir.
Sure,
yeah, if you have if someone has a burning question, you can ask. Otherwise, please leave.
So I didn't understand why, if the distance between the molecule is less than 1 hour's distance, there will be a possible repulsion between the nucleus of the atoms. Why it does not happen in this case? Why they attract?
Why do they attract beyond a van der Waals interaction?
Yes, sir. There's a
Because
repulsion
there is
between the
overlap.
nucleus of the.
Because see, van der Waals. Smruti, you can stop the recording. I think we are over the class. This is just off records. You don't have to record. And
Okay,
you
sir.
guys can carry on. I mean, Smruti and Sarah, just carry on. Okay, it's 12:25. Okay, see.